Okay, once again, hello everyone. I'm very honored to be here today, and we will talk about integrated omics approach for crop improvement. Can you hear me very well?
Yes, we can hear well. Thank you.
Okay, nice. So I will give a little bit of context, and then I will we will discuss about the multi-omics core concept, and we will focus on a specific case, and I will give a summary at the end of this talk. For the context, I would like to start with this press release from the International Livestock Research Institute dealing with the effect of climate change in Africa. As you know, many crops foundational to African diets, such as local vegetables, maize, sorghum, millet, will struggle to survive with this rising temperature. And under warming of 2 degrees C, crop yields across sub-Saharan Africa will decrease by 10%. And the warming beyond the 2 degrees C mark will cause crops yield to fall by up to 20%. And as you can see here on this map, African countries are most exposed to climate change adverse consequences. So according to the International Food Policy Research Institute, West Africa is already experiencing rising temperatures, shifting precipitation patterns, and increasing extreme events. This will be devastating for the Africa's food security, as those local vegetables, cereals are primarily very important crops in most African diet. This situation is already alarming in some African countries, as you can see here on this map, showing the global hunger index following countries. And we can see that the continent is in alarming situation with regard to food security. This is also confirmed by the FAO report that released both food security and hunger maps. And you can see here that Africa is not in a good position. So in this context, what should we do as a plant breeder? I think we should develop efficiently and in timely manner novel genetics resources that could be able to deal with both abiotic and abiotic stress. And we should also consider to develop high quality nutritious food that will cope with the food security issue. So the next question will be, how could we do that in time-saving manner? How could we achieve this goal? So this question will lead us to the second section of my talk, dealing with the multi-omics concept. I don't know if there is some question here. If no, I can start the second part of the talk. Can I continue, or do you have some questions?
Once you can continue, we shall have a question and answer session at the end.
Okay, great. So, to understand cell biology, scientists developed some concepts, based on the central dogma of molecular biology proposed by Francis Crick in 1957. And the central dogma of molecular biology is fundamental concept that describes how genetics information flows within cells. It states that DNA carries the instructions for making proteins, and this information is transcribed into a molecule called RNA. And the RNA then serves as a template for protein synthesis, where amino acids are assembled in a specific order to create a functional protein, which are the building blocks of life. And the final result is, of course, the phenotype of the organism, which what we can see and utilize in crop breeding, depending on the trait of interest. So the key question here is, how could we apply the recent technology to each compartment of the molecular biology? Let's remember that the central dogma is, briefly, DNA is transcribed into RNA, and the RNA is translated into protein. So at the DNA level, the omics concept is called genomics, while transcriptomics and proteomics correspond to RNA and protein compartment respectively. So all are interconnected and are extremely useful to dive into the understanding of plant biology and finding solution to mitigate climate change effect. So let's start with the genomics. So what is the genomics? Genomics is the study of an organism's entire genome, including the analysis of its genes, their organization, their function, and their interactions. It involves techniques such as DNA sequencing to decode and analyze the complete set of genetic instructions encoded in an organism's DNA. So as you can see here, I present some instruments that are very useful to sequence the genome of any organism. So you can see here, PacBio and Oxford Nanopore, two leading companies that provide long read sequencing services. And we have also Illumina, MGI, and Element Biosciences. And there are raising companies nowadays that are on the DNA sequencing market nowadays. So briefly, genomics helps improving crop yield and quality by and also tracing evolutionary relationships and studying the impact of genetic variation on traits. So when it comes to transcriptomics is a field of study that focuses on analyzing and understanding the complete set of RNA transcripts produced by a cell or an organism known as the transcriptome. And transcriptomics involves techniques such as RNA sequencing to identify and quantify gene expression levels, splice variants, and non-protein RNA. So application of transcriptomics include like identifying differentially expressed genes between conditions or tissues, or studying gene regulatory networks, discovering novel RNA molecules, understanding cellular response to stimuli or disease, and providing insight into development processes and biological pathways. So transcriptomics basically played a crucial role in advancing our understanding of gene expression and its regulation, helping in various areas of biological and medical research. And to do this, RNA sequencing, we have the possibility to do on the tissue, using the general pipeline here, described here. Or we can also dive more precisely into the cell level by performing the RNA sequencing on at single cell level that provide a high resolution to get more precise information about the gene expression. And here I will talk about the quantitative proteomics. Yes, it's also possible to quantify the protein. So basically, quantitative proteomics offer a good path to assess the relative change in protein expression while comparing, for example, disease-resistant and healthy plant. So it uses liquid chromatography-based separation combined with high-resolution mass spectrometry to identify proteins from biological sample, then quantify relative change in protein expression in 2 or more biological states. It helps to know the expressed protein and possibly the protein network involved in the defense mechanism, for example. But as we cover the DNA, RNA, and protein, there are other omics concepts that I should share with you today. So we all know that in chromosomes, DNA is wrapped around proteins called histones. And those histones play a crucial role in gene transcription regulation by helping to turn on or turn on off genes. So when certain histone modification occur, like acetylation or methylation, for example, the chromatin becomes more condensed, making it difficult for the transcription machinery to access the gene and the resulting resulting in gene silencing. But in contrast, when the histone modification under different modification, the chromatin structure becomes more open, allowing the transcription machinery to access the gene and leading to gene activation. It's like somebody is squeezing your neck, so you cannot speak well. But when your neck is free, you can raise your voice. So this concept is called epigenomics or epigenetics. So why this concept is important in crop science? In crop science, epigenetics plays a crucial role in understanding and manipulating plant traits, such as growth, development, stress response, or even disease resistance. So by studying epigenetics modification in crops, a researcher can uncover mechanisms that regulate gene expression. A researcher can also explore the effects of environmental factors on plant phenotypes and potentially develop strategies for crop improvement and breeding programs that can enhance yield, the quality and resilience of the crop regarding biotic or abiotic stress. Another concept I would like to share with you is called metabolomics. I think we all know some of these crops here, like the caffeine. We heard about garlic, black pepper, and strawberry, but they all have a distinctive characteristics. The coffee, for example, is a biostimulant with its own aroma. The black pepper is also used in food seasoning, the garlic as well. They have the specificity. And the specificity of these plants is mainly related to molecule called metabolite. Like the caffeine is one of the metabolites in coffee. Anthocyanin is that metabolite that play a crucial role in coloring fruits like strawberry and so on. The aline and allicin for garlic is the key metabolite from this plant. And in the black pepper, the pepperin is the key element that gives this specificity to the black pepper. So those molecules are called, I said, metabolite. And these metabolites are products and intermediates of molecular machinery that are catalyzed by proteins. So the change in protein expression can directly influence the metabolites produced. And the omics concept related to this metabolite is called metabolomics. So what is the utility of metabolomics? Metabolomics provides valuable insight into the metabolic traits of plants, helping in crop improvement and breeding programs. It's enabled to the identification of metabolites associated with desirable traits such as yield, nutritional content, flavor, or stress tolerance, facilitating the selection of improved crop varieties. But metabolomics also help in understanding the interaction between crop with his environment. I mean by environment, one side is about abiotic stress, so how the plant is reacting under heat, cold, drought stress, flooding stress as well. And also is metabolite also help by analyzing or identifying the metabolic change induced by microbial colonization when it comes to biotic stress, and the metabolic compounds involved in mediating beneficial as a symbiotic relationship or even pathogenic interaction when it comes to the attack of pathogen or any kind of biotic stress. So metabolite also play a crucial role in plant defense as well. And the last, not the least, is phenomics. Yeah, so when it comes to the biological phenotype, the omics concept here is termed phenomics, which is the study of plant traits or phenotypes at large scale, grouping the measurement and analysis of various observable characteristics covering the plant morphology, plant physiology, plant development, etc. So plant phenomics aims to understand and quantify the relationship between the plant genotypical characteristics, providing a valuable insight into plant growth, performance, and response to environmental stimuli and interaction with biotic and abiotic factors. Phenomics employs a large panel of platforms in field, indoor, imaging tools, and generate huge amount of data, which could be analyzed efficiently using machine learning approach. So altogether, the central dogma has evolved. We do not have only DNA, RNA, and protein, but we also have epigenomics or epigenetics. We have metabolomics, and we have as well phenomics. But it's also important to draw your attention that this list is not exhaustive. Of course, we have new omics concepts coming or emerging nowadays, such as lipidomics, for example, focusing on lipid. Yes, this is just one example. And one key thing is all these omics concepts generate a vast huge amount of data and require mathematics skill for their analysis and interpretation. So then we move to the third part of this webinar by asking a very important question: how can we integrate those multiple omics concepts for crop improvement? So we try this during our PhD journey, and we focus mainly on sesame. The scientific name is Sesamum indicum. So Sesamum indicum basically is known as the queen of oil crop. And the price of importation was estimated to more than 2,000 million USD in 2017. And most importantly, there is one component in sesamum called lignans. Lignans are specific specialized metabolites in sesame that play a role as antioxidant and is very useful in drug industry. So when we focus on this lignan, the market is estimated to more than 300 million US$ in 2020, showing the importance of this metabolite in the world. And The first step I would say is to know your genetic resources. In order to integrate those omic concepts, it's very important to know what you have as genetic resources. So, do you have a wild species? Do you have a land race? Do you have a modern cultivar? Or you have a core collection, or you have a mapping population, and your species is native from your area or is introduced? What is the biological status of your material that you have? This is very important starting point that will be very useful for further steps. So when we know that, it's also important to check your materials. So when I guess you receive like some resources from GeneBank, it's important to go to the field and check them and see which plant is useful for this target of for the breeding programs. So we conducted field phenotyping, a field experiment on a worldwide collection of sesame, over 500 accessions. And we studied 18 agronomic traits and 6 most focus on seed quality like oil, fatty acid, and most importantly, sesamine and sesamolene. Sesamine and sesamolene are those lignan, very important in sesamine, that is very trendy now in pharmaceutical industry. So we set this experiment at 2 locations, and we use augmented block design with 8 replication. So here is the output that we got. So we can notice here that we got a quite rich diversity in terms of flower color, diversity in terms of seed color, diversity also regarding the agronomic important trait, which is the number of locule per capsule. So here you can see that we have some capsule with 4 locules, others have 6 locules, and very interestingly, 8 locules per capsules. So meaning that if we are interested in improving the number of seed per capsule, we might be interested to target this accession that produce 8 locule, that have 8 locule per capsule. And interestingly, after clustering our accessions, we found that even though we were in Korea, we found that accession from the northern part of Africa and eastern part of Africa, they contribute highly to the yield, even though we were not in Africa, but in South Korea, they adapt very well. So mainly those accessions were located in the cluster 3 here. So they can be used as parental lines regarding the yield improvement, for example. And following the fact that we could not use all the 506 accessions for genomic study because the cost is very high, we decided to make a core collection, and we were able to reduce those 506 accessions to 102 accessions actually, by keeping the geographical and the phenotyping diversity as well. So we were able to pick up, to find out some specific accessions that I marked here, that are rich in fatty acid, very rich in lignan, like sesamine and sesamolene as well. So these materials are actually under assessment for developing mapping population. And when you have your genetic resource and you try to do to check what you have, you might be interested to, okay, how can I use it? How can I incorporate the omics stuff here? But before moving forward, we already covered two concepts, two important concepts. First is phenomics, and the second one is metabolomics, because we try to quantify metabolite in every accession that we used for our phenotyping. So the next step is to do genotyping, or even whole genome sequencing. To do basically genotyping, we have two main approach based on the technology. We have the array based genotyping, and we have also the NGS-based genotyping. So the array base is the older one, and the oldest one, and the platform are Affymetrix Axiom, Affymetrix GenChip, Illumina Infinium BedShip as well. But I noticed that recently people are using GBS because maybe the cost is quite affordable. Others are using DartSeq or RedSeq, DDRed, RedSeq as well. So the project found will influence the type of sequencing platform that you will be using as a plant breeder. So it's also possible to sequence the whole genome instead of sequencing like target sequencing. It's possible to sequence the whole genome completely. And it's also good to have more than one reference genome. So if you have multiple genomes, high quality genomes, that's called span genome, you can be able to try to see the structural variation among those genomes, and try to see the consequence of this SV on the phenotype as well. And it's also important to mention that at this stage, it's possible to do the genome-wide association studies, like to find out gene of interest, for example, the gene that you are interested in. So to find out this gene, you might need some resources. You might need, like, you need a map to find out your way. If you are new in a new environment, you need some orientation. Well, so where to find the gene of interest, and how we could find the gene that we are interested in. So the step 2 is about to generate the genomic resources, like database or gene, or genome assembly, or some molecular markers, some QTL. We need this information to accurately find a gene of interest. So in our study, we were interested in oil and lignan as well. So we pick up one accession, which is Sesamum indicum cultivar Gombe. Gombe can produce more than 1 ton per hectare. And the oil content is quite high, around 50%. And most importantly, sesamine and sesamulin, those lignan, quantity is quite interesting for us. So we decided to select this plant for a whole genome sequencing project. And then we used for that a combination of sequencing technologies like Illumina, short read, and we use also PacBio long read data, and Hi-C as well, to reach a chromosome scale genome assembly of Sesamum indicum cultivar Gombe. So we were able to reach 13 chromosomes, and we annotated this genome in order to get our map that we need to find novel genes. And the data for this assembly is already available in NCPI platform. So At that time, colleagues from China also produced several whole genome sequencing data, like UZ11, Mishuzima, Baizima, Suita from India, Neizongzi 13 from China as well. So we were interested in what aspect is quite special for Gombe when compared to available data sets out there. So by performing a comparative genomics, we were able to find out some specific gene families that basically are involved in fatty acid biosynthesis, involved in lignan biosynthesis, as well as oil biosynthesis. So this result give us important insight about the choice of the material that we selected for our breeding purpose. So once you get the map, the genome, it's now time to investigate your gene of interest. And since we were interested in lignan biosynthesis, following the pathway, we were interested at upstream level of the pathway. I mean, here, colored in yellow, we would like to know which kind of gene are involved at early stage of the biosynthesis. So to reach that goal, we use transcriptomics approach by selecting seeds like white seed versus black seed. We compare as well seed with rich oil versus seed with low oil content. And also we use RNA sequencing following organ like a root, leaf, seed, etc. And we try to use all these huge data to find out genes that are possibly involved from the at the upstream of this pathway. And mainly we're looking for two kinds of genes, peroxidase and lacase gene. So using this huge transcriptomic data, we were able to point out here in red star some critical genes that pop up from our analysis. So mainly we found two peroxidase and two lacase genes, and we checked them using qPCR. And now we are trying to do a functional validation, and we are planning to do hairy root methods to improve the lignan production in sesame. But most importantly, we need to link up the transcriptomic data with the pathway dynamic as well. So since we are at the upstream level, we were expecting like our gene should, the expression of our gene should go up here and here, prior to see this one in red, which is the pino-resinol content, the first following a molecule after the first step of the pathway. And we see that in both cases, when the pino-resinol increase, the gene expression is already down. So meaning that the selection should also take into consideration the dynamic of the biology of depending on the topic of interest. So it's very important to design your RNA-seq experiment accordingly and to follow as much as possible the biology of the species. So until now, I just share experience about what I have done so far. But in the same time, scientists from China, they also use whole genome data set here, and they perform a genome-wide association analysis on several traits, including root number, root dry weight. And they were able to find, some locus with a high variance regarding a specific SNP. And that they test using Arabidopsis thaliana plant model. And basically, what they found is that they found key genes that is responsible of root biomass. So why root biomass? Simply because root system plays an important role when it comes to handling global warming, for example, when it comes to drought stress. So root is one of the key points that is a variable to mitigate drought stress. So this finding is very important in sesame field that could help to boost the crop, to develop novel crops with a high ability to deal with global warming scenario. And when the gene is already detected, now it's time to move into research and development section. So in the sesame field, there are already huge amounts of seed available on the market that people are, buying for food, for seasoning, for example, and for many others industry, like in the healthcare, drug industry, cosmetics industry, in food industry. They are full of options for sesame. And to show the importance of this crop, there are several patents already available in Taiwan, in US, India, in Japan. So people are actively working on sesame to find a novel application in a different area of our daily life. In Africa, because this is where we are, in Africa, sesame, for example, in Senegal, sesame field now is about like in promotion now. So people are trying to grow sesame as a secondary crop in the field. And in Ethiopia, the system is becoming like more technical and industrialized. So they do have some machine for seed treatment as well. And what is very nice on the continent is 3 years ago, the continent is the most contributing continent regarding the production of sesame seeds in the world, with Sudan as the leading producer of sesame seed in the world. So this area or this crop is a promising crop to boost, the economical sector in Africa regarding agriculture. So I discussed about sesame. So, but there are other cases, for example, for rice, just last month, the scientists from UC Davis University of California, in collaboration of Huazhong Agricultural University in China, they released a cultivar of rice called Kitake. Kitake show high yielding capability as well as resistance in blast. Blast disease is one of the devastating, disease for rice in the world. And scientists were able, using the genome editing technology, to generate cultivar that not only provide a good yield, but also is resistant to rice blast disease. Another example is the well-known golden rice. This rice specificity is they have beta-carotene gene that is a precursor of vitamin A, which is also important for nutrition aspect as well. So we also have as an example the development of a drought-resistant maize by Pioneer and Cultivar AgriScience. So using the CRISPR-Cas9 technology, they were able to design a maize varieties that can produce good yield under drought stress conditions. So all these examples just to show that it's possible to quickly use the genomics, omics concept globally to do crop improvement. So as summary, in our study, focusing on sesame, we use genomics because we generate the whole genome, the gene content, and so on. We use transcriptomics as well, metabolomics, and phenomics to find out novel genes, to find out good accession that will be used to develop new population and find out novel genes regarding yield, regarding the oils, and regarding also the lignan biosynthesis. The take-out message today is we as a breeder on the continent, we should consider using omics because omics enables fast forward breeding and can be very useful for food security. And to reach this goal, the genetic diversity is the key element. So the wide accession that we have, or the land races, most of the time rich in genes like resistant genes or genes related to yield or productivity. So we should develop some gene bank on the continent that can gather these important resources that is needed to improve our crop on the continent regarding the production, but also regarding the quality of the food that we want to get. Of course, using omics will lead to a huge amount of data, and it requires to form or to train bioinformaticians on the continent, and also to do to include as well a machine learning approach to efficiently manage those data. And last but not least, this is a key point, that is the gene editing stuff, which is now under debate. But I heard last week that the EU Commission, the European Commission, they are trying to relax the GMO law, and maybe soon in near future, if the law passed, we could see in the European market the gene-edited crops over there. So I provided a graphical abstract that will be shared to you guys, summarizing all the concepts that I've discussed and that I've shared. And with that, I will say thank you for listening to me. And I'm happy to have your question, to answer your question, to receive your contribution. And last thing, yeah, you can scan this QR code. You will be able to get the presentation. Thank you very much.
Okay, thank you, Ange, for this interesting talk. Hi, everyone. My name is Fréjus Sodeji, and I will be facilitating the Q&A session. So first of all, I would like to thank Ange again for this interesting talk, taking us through the concept, the various concepts of omics. And what I like most was the case study. We showcase of how we could apply this different component of omics in order to improve having good varieties to address different challenges on our continent. So without wasting time, I think we have a few questions in the chat. Ange, I believe you can see the chat. Otherwise,
I don't see the chat.
Okay. So let me read them out for you.
Okay.
Okay, so there is one question from Valerie. She said, very interesting research work. Thank you for this. My question is, what kind of software is used to generate all the data, including the chart and pictograms, and how these data are interpreted to come to your final conclusion. So that is from Valerie.
Yeah, thank you, Valerie, for this very nice and interesting question. Sure, as I said, Omics data means big data, you know, and to deal with big data, we need to have bioinformatics skills. And how I was able to assemble and annotate genome, it's a process that requires several softwares and several encoding skills that I have used to assemble and even to find out gene of interest. So I could not give you clearly, I cannot explain all the process now because it's quite long and depending on the step we are. But basically, to find out the gene or to construct a genome assembly, there are a bunch of tools out there that is possible to use, depending on the type of data you have. If it is a short-read data, or if it is a long-read data, or high-speed product map data, if it is transcriptomic data. It's case by case. Depending on your data, there are subsequently appropriate tools that is possible to use. Maybe I can share maybe a link later on in the mail showing that a website that you can go there and then you can see the different type of tools that is possible to use depending on the technology. Yeah, that's what I can. I don't know if I answer Valerie.
Yes, thank you very much.
Yeah,
you're welcome.
Thank you, Ange, for that clear elaboration. So the second question was is from Aminata. She would like to know the difference between omics and the GM plants.
Okay, the difference between omics and GM plants. Okay, omics, like genomics. So in genomics, we have genome and omics. As I said, omics just means big data, and genome is the DNA, actually, basically, to make it simple. So every omics concept deal with a specific aspect of the central dogma of molecular biology. If it is genome, RNA, protein, metabolome, or epigenetics, whatever. And GMO is just genetic engineering techniques that will utilize the information from the omics to develop novel crops regarding, for example, disease resistance or regarding simply productivity. But in that GMO, precisely, we do have now the gene editing techniques, which is now something that some countries in the world, they accept that and already implemented that in Japan, for example. But some other country or continent like EU, they are still, reluctant to use that because we don't have enough data to see the consequences of using gene editing technology. But for sure, gene editing technology is offering a very excellent, very fast way to edit a specific gene without, wasting too much time, and very effective to design crops regarding disease resistance or about stress. So the omics is just the concept, the instrument that I use to dive into a specific a path of the central dogma, and the GM is just the genetic engineering side that is useful to design novel plant that will be used to answer a specific question. That's what I can say.
Okay, thank you, Ange, for that question, answer. And there is a question from Wilfried. I think you may give a little bit of clarification here, because he was asking, in the Sesame case, you presented Manhattan plots showing GWAS result from two different root threads as an indication of a differential expression of genes. Did you manage to identify the exact genes that were differentially expressed using the result from GWAS? I think you need to clarify that.
Yeah, sure. I think there is something that we need to clarify, of course.
Yeah.
The GWAS is not for differential expressed gene. The differential expressed gene is about the transcriptomics. So we use transcriptomics to find which gene is expressed in which condition. But regarding the GWAS purpose is to find out, some loci, which region in the genome harbor a key gene of interest. And here we are not interested in differential expressed gene, but we are interested in a specific SNP peak that will guide us to a locus of interest associated with the trait that we are interested in. For example, here the trait is root number, and here the trait is root dry weight. So this SNP seems to be highly related to root number in Sesame, and this SNP also seems to be related to root dry. And interestingly, those two SNP are located to the same chromosome here, chromosome 15, I think. Yeah. So this root number and root dry weight are important traits that will determine the ability of the plant to mitigate the global warming scenario. So by doing GWAS, we can find out putative locus, but this locus, this loci should be validated experimentally to make sure that what you found is usable. Yeah. So the GWAS purpose is to find the fit regarding the of interest, and the analytic or transcriptomics approach is to find out which genes are differentially expressed in which condition, depending if it is a disease or not, between disease resistant and disease susceptible plant. I mean, I don't know if I clarify those two concepts clearly.
I hope Wilfried is listening, and yeah. So we have other question from Dr. Ba, I believe. He is asking, he wanted to know how long it took you and your team to draft the first reference genome of Sesame.
Oh, that's a very nice question. To answer correctly to this question, from the DNA extraction to the chromosome scale genome assembly, it took almost 8 months. And I will explain why. It could take less, but 8 months in our case, simply because I faced some issue during the DNA extraction step, because we wanted to use not only short read but also long read data, and long read data require a high molecular weight DNA, and that DNA should not be, should be intact as much as possible, not fragmented. So Sesame is rich in polyphenol content, which makes it difficult to extract the DNA. So we took almost 2 months to calibrate our DNA extraction protocols And once we extract that, we send it to sequencing platform, and we perform the long read sequencing platform. And we also perform a high-C contact map in order to help us to reach the chromosome scale. So globally, it took 8 months, but it depends on the plant, depends on the challenge, because each project is different. It depends also on the ploidy of your crop of interest. My plant of interest was sesame, and it's diploid actually. Sesame medicum is diploid, so it's more or less easier to handle diploid genome. But when it comes to polyploid, it's much more difficult to assemble it accurately. But thanks to the improvement of long read, now it's possible to, to perform haplotype resolve assembly, even for polyploid. It's still challenging, but with the current state of sequencing, we can do much better. Yeah, so to answer the question, 8 months, but this is not, the route that you should consider, because it depends on the project, depends on the plant specificity, it depends on the plant ploidy, depends on the plant heterozygosity, for example. Yeah.
Thank you. I think we have, because of time constraint, I think there is one hand up. Edwin, if you would like to ask question to Ange, you can unmute yourself and speak directly.
All right. Thank you very much for the presentation. I really enjoyed the talk. Yeah. What I wanted to find out is that there are actually a series of questions that I wanted to ask on the Sysmin project, especially with the transcriptomics. What I wanted to find out is that first, did you with a gene that was identified, did you have any prior knowledge that probably maybe the gene was having an additive effect or anything before you went ahead to do the transcriptomics to identify that gene?
Yes. First of all, we studied the pathway first, and there are previous works before my project, and we do have an idea of what's going on in the pathway, and we were suspecting these two kind of gene, peroxidase and lactase here, that might be possibly involved at upstream level of the pathway. So from that knowledge, we decided to use huge data regarding different facets of the seed color or oil content or organ wise to find out if this kind of genes are present. Even we use also the whole genome data set from 5 sesame species. Yeah, so we do have some knowledge before jumping in this transcriptomic approach. Sure, it's very important. Doing transcriptomics require some knowledge about the biology. Yeah, it's very important. If not, you may lose your time, your budget for something that might not be what you want.
Yeah, and thank you very much for the clarification. And I also wanted to find out, since you did the whole transcriptome sequencing, did you also try to do some sort of gene interaction, maybe using a network approach to find out if there are other genes that may be involved in the pathway, but not necessarily heightened in the differential gene expression?
Actually, no, we didn't reach this step, but the reason why we didn't perform this is just because of this work have been done actually by other scientists. So we had this information earlier. But what we did is we tried to see which transcription factor might be related to this specific pathway. And we do find out by doing bioinformatics analysis, some TF like ORCID, for example, that we found as most prominent during the analysis. But once again, it's just analysis. You know, we need to confirm that technically to validate that to see if really it's impact on the pathway or not.
Okay, thank you very much.
Yeah. Anyway, the paper is available online, and feel free to check it. Yeah.
All right, sure. I'll do that.
Okay.
Yes. Another question?
Yeah, I was trying to check with you if you still have more time for questions, and then if you are willing, we can continue with some of the
sure.
in the chat. Okay. So there is one question from Carlos. Of course, he said that please let me come in from another perspective. Above all you have said, one important question remains to speed up the integration of omics in plant breeding program in sub-Saharan Africa. So the question is how to leverage the access to and use of all the omics platform to African breeders. So if you could elaborate a little bit on that.
That is a political question, actually, I think. First of all, I think we need a platform. We need platform. We need sequencing platform as well. So as far as I know, we used to do most of the time outsourcing. We used to send our samples to outside, like in Europe or Australia or whatever, in US, to perform genotyping and so on. So I think we need to have on the continent, in West Africa, precisely, you know, those platforms that will be available with trained people, with bioinformaticians as well. And in such that condition, we could efficiently and quickly implement omics in our context, dealing with the troubles or dealing with the issues that we have regarding food security issues. So the first point is to have those platforms available. So I would like suggest for institutions that are involved in the promotion of agriculture in sub-Saharan area to try to promote the acquisition of those sequencing machines, for example, and to set a lab, a genomics lab, that will focus on the crop of priority from sub-Saharan, like millet, like sorghum, like rice, whatever, so that we can practically, use, we can take advantage of omics to solve the problem on the continent. But once again, is this is most of the time this is a political decision, and also business, because those companies are trying to set up on the continent nowadays. So in South Africa, I do know that we have a backfire company services in Inkaba. I don't know if you know Inkaba. Inkaba is present in South Africa and in West Africa as well. But the PacBio sequencing machine is available at South Africa now. So we do have also one PacBio machine in Mali for health purpose. Yeah, so I think nowadays some countries are interested in omics, and they are trying to buy the machine, trying to set up lab to deal with our issue on the continent. So that's I think the key point. We need to bring on the continent those instruments that is useful to dive into agronomic interest.
Okay, thank you. So there is another question from Ademola. He wanted, I think the question is, do we need a large data set before omics can be used? And can omics tool be used in a mapping population and identification of genes in fusarium with disease in banana? So
yeah, sure. As I said, omics is just how can I say? It's just the concept actually, you know, and of course there are several cases using genomics, even precisely transcriptomics for banana cases. I know there is a scientist from IITA who is working actively on banana using transcriptomics approach. Her name is Lina Tripathi. Yeah, Lina Tripathi. So she and her team, they developed a transcriptomic approach. They used transcriptomic approach actually to find out gene expression by comparing resistant material versus susceptible plant as well. Yeah, so it's possible to do. It's already ongoing, even on the continent at IITA. Yes.
okay. So there is a question from Paul. He's asking which indigenous crops have benefited most from the application of omics in West Africa.
Indigenous crops. The word indigenous is quite interesting. But for example, I will take this example of West Africa. And you know that in Senegal, for example, millet, sorghum, very important, certainly in area with high drought stress. So actually, a big project led by IQSAT, Dr. Rajiv Vashni, they use genomics, transcriptomics, in order to first sequence and a good quality of sorghum and millet genome. And they are promoting highly nutrient, millet and sorghum. So nowadays they are targeting genes that are related to, for example, iron accumulation in millet and the sorghum. So yeah, so things are going on actually, and the results of this work will be highly valuable for the continent as well. And there is also a recent publication of the genome from a collaborative work with GBIOS from Benin and Netherlands team. They were able to assemble the genome of a crop specific to West Africa called, if I'm not wrong, Cisre or something similar like that. So even though we don't have the all infrastructure, some scientists are collaborating with some institutions to bring on the continent omics work. And at the end of the day, I think we are coming to that point that soon we will see sequencing technologies companies coming to Africa to develop their business. Yes, so to answer the question, yes, there are ongoing projects regarding Sub-Sahara, and hopefully we can see the result of this research soon. Okay, Regis, it's okay? Oh,
Regis, I think you are muted.
So yeah, so I was reading another question from Romuald. He was asking between the omics breeding and mutation breeding, which one is faster? So
for sure, mix.
okay, so
by mutation, I think this is techniques of bombardment, something like that. So I think omics is, yeah. But of course, this is a general question. It depends on what material you have, what you are doing, and the pre-knowledge that you have. Yeah, it's case by case.
okay. Okay, so with that, I would like to thank you, Ange, for the answer to the question as well as the clarification about some of the comments which were made in the chat, and also I thank the participant for making this session very interactive and interesting. And it's worth to state that this is our second webinar in the series of the webinar we are putting up with in the coming symposium, because we will be having a symposium, I believe, on omics. So this is to lay the ground. So if you are interested in a symposium in omics, please, you are welcome to join the symposium and submit your abstract. So with that, I will over to you, Raquel, please. I think you are muted, please.
I did it too. Thank you for that. Sorry. Thank you very much, Regis, for leading the Q&A. And thank you to everyone for joining us and for your participation. I think we had a very nice discussion about omics from multiple points of view and multiple applications. A very special thank you to Dr. Ange Zoclanclounon for your informative and insightful presentation.
Welcome.
As Frey just mentioned, we do want everyone to be on the lookout for information coming soon about our upcoming 2023 symposium. So thank you again and have a good afternoon.
Thank you. Bye-bye, everyone.
Bye. Bye.